This is TWIS. This Week in Science, episode number 790, recorded on Wednesday, September 9th, 2020. How the West Was Burned. Hi, everyone. I'm Dr. Kiki, and tonight on TWIS, we will fill your head with pain, bodies, and genes. But first... Thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Whatever you're doing right now, wherever in the world you might be, keep an eye out for something interesting. Listen to a sound you might not have otherwise paid attention to, or some detail in your environment that went unnoticed before. Look and listen. Observe the world of nature. Life in all its many forms evolved over billions of years, overcame every possible earthly obstacle to exist. What could you learn from that? Look and listen. Observe the world of technology, accelerating our evolution with the first stone tools, giving us the cutting ability of sharp claws that nature had overlooked. And lastly, with the ability to adjust the genome itself, influence, if not control, our evolutionary future. Look and listen to the subtle creatures and objects around you, and know that it's connected, all connected to observations made long ago and being made now. Observations yet to be made by generations yet to come. And all of these observations and noticings of things, when backed up with a little bit of knowledge, connect to that first mitochondrial Eve with you, and you with This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. 790, 10 away from 800.
You know what we should do for
Too soon.
episode
Sorry.
800 is we should probably all get together and do something really fun. Oh, wait.
Yeah, I had all these plans for a episode 800/20 years of TWIS kind of get together. And then I went, this year is just a do-over.
Wait, why would
The plans will happen. We'll make it happen. Internet togetherness is a thing.
That's
Yes.
fine. It's fine that way.
It's fine that way. Everyone, thank you so much for joining us tonight. I hope wherever you are safe, and you are, I hope that you are safe. The West is on fire. There are so many fires right now, and they're growing and they are combining, and towns are being burned, homes are being lost. And I did joke just a moment ago, but it is a very, very serious situation. And part of it is climate change. And the news has not really been discussing the fact that climate change is responsible, in part for the heating, for the odd wind situation that we have up here in the Pacific Northwest that is fanning the flames. Climate change is a definite part of the increased burning of the West. And so that kind of information is very important. So I urge you to, if you haven't yet, after all our conversations on this show, maybe take a moment to think about how climate change is affecting your local environment.
Yeah. So, I mean, climate change doesn't cause fire, but it makes fires more common and burn longer and hotter and farther. Especially with drought, it burns faster. It burns faster, it burns farther. So it's definitely, I think that's why we don't hear a lot about it. It's in the nuance.
The nuance. Well,
People
what
don't
it
want to
is,
get into the nuance in the news.
nuance. That nuance is like speeding up desertification of California is what it is. So we have science coming for ya. I have science about, what did I bring? I can't find my little cube that tells me stories about stress. I have frigid birds. They're very cold, and milk drinking because it's something I can't really do. What do you have, Justin?
I've got fish that can walk, a new ancient ape discovery, what makes people from Vermont the toughest in the nation, goose eggs in the search for alien intelligence, and what biker rallies and church leaders have in common.
Oh, very interesting. All right, commonalities. It's where you start conversations. And Blair, what is in the animal corner?
Oh, I have dead bodies. I have naked deaf animals, and I also have endangered species.
Well, thank you for bringing the fun.
Always.
That is TWIS. If you have not yet subscribed to our podcast, you can do that anywhere podcasts are found. Look for This Week in Science. We are also on YouTube and Facebook. And you can find our website at TWIS.org, twis.org. That's where our website is with all sorts of information. But now, time for science. Quick science summaries. Okay, with all the stress lately, do you feel like sometimes you just can't sleep? Like
Yes.
You're like, I'm so stressed out and I can't sleep and I wish I could sleep.
Yes.
Yes.
Yeah. That was me on, what was it, Monday night? I barely slept.
Oh, no.
Yeah. I don't know why.
And it doesn't help anything because it just kind of makes it worse. Why? That's what researchers have been wondering. And we know it starts in the brain somewhere. And researchers decided to look at the neurons in the brains of mice. They looked at the neurons in an area of the brain called the lateral hypothalamus and also looked at what are called the paraventricular nucleus of the hypothalamus, where corticotropin releasing hormone neurons live.
Oh.
Yeah, what they found is that when the mice were restrained, that's a known stress function on mice. When they were restrained, the neurons that are in this area of the paraventricular nucleus of the hypothalamus, the corticotropin releasing hormone neurons, got active. These corticotropin releasing hormone neurons, they released corticotropin releasing hormone, which is a hormone that goes on to lead to the release of cortisol, the stress hormone that we know of. Additionally, they signal to hypocretin neurons. And these hypocretin neurons, they kind of elicit a hyperarousal. And anyway, the end story is they have found the neurons in the mouse brain that keep you awake at night when you're stressed out.
So just burn them off, right?
Just get rid of them.
Well, actually, when you say that, but you say that, it was like it's a joke, but maybe there is a way to, like, earlier in the day, induce that stress, release those neurons already so they're gone before you go to bed.
Maybe that's exercise.
Maybe burning them off.
A nice sleeping cap that just, electrocutes that area. Just
or genetic modification.
Oh,
in the case there's that. In the case of these mice, genetic ablation of the neurons that are involved, or a knockdown of the gene, actually taking the gene out, making sure it stopped working, counteracted this restraint stress-induced hyperarousal. So if you get rid of the genes, you get those neurons so they're not activated, the response goes away. You can go to sleep at night. Basically, they found that when they did that, they ablated the neurons or got rid of the gene in the mice. When they restrained them and stressed them out, they still slept like babies. It was just fine. It was just fine.
But what you're not bringing up is that what they also found is that the mice found it had a tendency to fall asleep while driving. They just
they're not getting stressed out at all.
They got tired. The urgency of the situation didn't cause any sort of stress reaction.
That's an interesting point. Yeah, you kind of need that.
It's there for a reason.
That's why the cap would be great, right? It's just like a switch. Like, okay, it's bedtime. Time to turn this off now.
What is
Yeah,
this?
maybe
You're
someday.
inventing magical hats now? What is
Yeah.
this magical hat?
No, someday it's just going to be Elon Musk's, little insert into
Oh,
your
then I
head.
just open my
That'll do it.
phone and go sleep mode.
Sleep mode. You go. Exactly. Yeah, they also connected this specifically to a change in the immune system. They found by looking at the peripheral blood that there was actually changes to immune cell distribution and functional changes in the peripheral blood related to those cell changes. So what they're finding is a connection between those neurons being turned on in the brain, the hyperarousal starting, stress and lack of sleep, and the immune system all being tied together. So it ties together a mechanistic loop for all the complaints that we have about getting stressed out and not being able to sleep, and then getting sick or not feeling good afterwards.
Yeah, I think somebody made a
amazing.
joke in the chat room, but I would be very interested to see what happened if they were dosed some marijuana.
Or
I don't
a
know.
glass
Maybe that
of
helps to
wine.
reduce that. Yeah, or a glass of wine. Exactly.
Or there's some warm milk. That's also a thing people.
That's what I'm saying.
We'll
Like
talk
now
about
that we
milk
know
later in the show.
what to look at, you could actually test a bunch of home remedies for sleep and see what works on that area of the brain.
It turns out, after much scientific study, that moonshine, white lightning, specifically, seem to contribute to
I don't know if any of these
night's
things.
sleep for a bunch of
Yeah,
participants.
I don't know if any of these things are going to be like specific to an area of the brain. They're going to be just like, and go to sleep now.
And
Yeah,
override.
it's more of the
General
shotgun
over-
approach, right? Just
Yeah. But maybe there is an approach that'll be more specific sometime in the future
yeah.
for all that stress. Okay, so from stress, let's move on to pain. Justin, do you have a pain story?
Yeah, so, okay. Actually, on today's date, September 9th, but 1776, the United States became the United States for the very first time, thanks to the Congressional Congress adopting the term officially. It had been in the Declaration of Independence a few months prior, July 2nd, and then published maybe July 4th, which is why it's kind of a big deal day. But it was still, at that point, the United Colonies. Not until the September 9th that we became the United States. And while the word united is right there in the name, America's rarely united around how it feels about anything. And it turns out that also includes how it feels pain. We as a nation of people feel pain differently state by state. At least that might be one thing you can pull from the conclusion of researchers from Texas A&M University and University of Pennsylvania, who looked into opioid prescribing practices across the United States. Specifically, they collected data from around 100,000 outpatient knee surgeries nationwide. And then they looked at prescription rates and dosage of opioids in those prescriptions. What they found was a very wide-ranging variability in both rates and to prescribing dosages. For instance, they found that prescription strength and number of tablets, dosage prescribed, was highest in Oklahoma out of all 50 states, and lowest in Vermont out of all 50 states. And Vermont was lowest in both number of pills and in dosage, and Oklahoma was highest in number of pills and in dosage. So very interesting. With Oklahomans being actually prescribed almost twice the number of tablets and more than twice the dosage or the potency of those pills, which could indicate that science, since people are generally expected to have somewhat similar experiences post-surgery, and that overprescribing can lead to addiction, that there's a need for a strong national guideline for prescribing opioids. That or some parts of the country are just tougher than others. But mostly it seems like the rural center, what you would call a flyover state, are being prescribed at higher rates. Yes.
Yeah, for the
and so the question is, are these people weaker? Do they have lower pain tolerance throughout? Or is there perhaps some association
Overprescribing happening. Just
between, yeah. There was, I think, a correlation between education and dosage. So the more educated, the less likely you might be to be prescribed opioids at the higher rates. So
interesting, but part of that is the person doing the prescribing, right? Like there's all this thing about how who prescribes opioids or overprescribes opioids target specific groups. So that is also part of this.
Yeah, there's definitely going to be multiple factors that come in. There's not going to be one easy answer for why, but I'm going to guess that overall there is overprescription happening, but why that overprescription is happening may be different in different places.
Yeah, so that's, I mean, I think that's also the point that they make in this study is when you look at this, pain shouldn't vary that much.
Right.
From Vermont to Oklahoma, it doubles in dosage, it doubles in number of pills for the same procedure. That shouldn't be, right?
I think we can debate this, but I mean, the bottom line is we don't know the answer for why. Mm-hmm. There is just a difference between states that is interesting. It deserves more study.
Mm-hmm.
Yeah.
Yeah.
Let's talk about plants. Dead bodies.
Dead bodies. So first comes stress, then comes pain, then comes death. So this is perfect.
Yes.
So longtime listeners of the show will know that I'm obsessed with the Body Farm in Tennessee. So this is University of Tennessee's Body Farm. It's actually technically called the Anthropology Research Facility. But it's where they put dead bodies and watch them in various states of decay in different situations. They look at what it looks like, what decay looks like, but also more specifically, a lot of the time they look at what kind of biota grows on and in a body in different states of decay. So that then forensic scientists can go and they can, if they find a body in the woods, they can say, okay, based on the types of maggots in this body, the shape that it's in, etc., this body is 3 weeks old. So then that helps with all sorts of kind of the detective work on the other side, figuring out who the person is and what the circumstances around their death was and all that kind of stuff. So that's really helpful. But for the first time now, a new article in the journal Trends in Plant Science is looking at plants and their role in the decomposition of bodies as pertains to finding missing human remains. So that's the other side of this, is that you can, if you see a lot of a particular kind of maggot in a particular area of a forest, you might say, ooh, something's dead around here. And that might help you find someone you're looking for. So this new kind of exploration is looking at how plants can help us find dead bodies. Normally, when people go out searching for a body, if there's a lot of tree cover, it's pretty difficult. This is an area that, you can't survey from a helicopter. Just doing a blind sweep is really, you're going to have a difficult time finding a human. But you can utilize tree cover in body recovery missions to your advantage. They posit by detecting changes in plant chemistry as a signal of human remains. So in this journal article, they outlined the steps needed to make body recovery using vegetation something that we could do in the near future. So they assessed, they're planning on assessing how cadaver decomposition islands, which is the zone immediately surrounding a human that has died, changes the nutrient concentrations of soil.
Yeah.
And how those changes manifest in nearby plants. So one of the most obvious things is you have a huge release of nitrogen into the soil.
Lots of nutrients in general. There's tons. Yeah.
And so that can change leaf color. It can change reflectance of leaves, which I think is very interesting. The problem there is another large mammal like a deer dying in that area could have a similar impact. However, the next step of this is to see how specific metabolites like those from drugs or food preservatives, things that we eat that deer don't, might have a specific influence on plant appearance. So not just a dead mammal, but a dead mammal that's been eating Twinkies. So
it won't find any vegans this way.
Potentially. So this is the other thing they said is if they know anything about the specific person they're looking for, and they know, for example, that they're a heavy smoker, they could actually use a specific chemical profile that would trigger a specific plant response, and that would make them easier to locate.
Wow.
So this is still pretty far out, but this is what they're hoping to get to. So they're hoping to develop imagers to scan plants for fluorescence or reflectance via drone so that they could see like a chemical signature, like, hey, something dead is down there. And so this is just so cool. They think it's still a few years away from using plants as search tools in body recovery missions, but this is a collaborative effort of botanists, anthropologists, and soil scientists, and they are starting out at this body farm now. So good.
Want to find a body? Ask a plant. All of us probably have a pair of blue jeans in our closet, if not one. Maybe a stack of them?
Yeah, I've got several, I think. Yeah.
Your blue jeans aren't just in your closet. They're everywhere.
What?
Yeah. Researchers looking at microfibers in the environment did sediment samples of the Canadian Arctic archipelago, the Laurentian Great Lakes, and shallow suburban lakes in southern Ontario. And in looking for these wonder fibers, they found that 21 to 51% of all microfibers in the sediments were from people, human-modified cellulose. And of those, 40 to 57% of the microfibers they found were indigo denim microfibers.
What?
Yes, over half, or around half of the microfibers they found are from our jeans.
But I thought jeans were made out of cotton, so it was okay.
It's cotton. They're microfibers. A lot of denim these days. It is cotton. Some of them use stretch fibers that come from more polymers. Bamboo, but that gets broken down and made into a plastic, and that's a stretchy, and then it's a polymer. And so some of these microfibers are not necessarily great, but what it gets at is not that the genes are bad necessarily, but they are an indicator of the pollution that we are spreading out into an environment. So they are, what, in the words of the researchers publishing in the Environmental Science Technology Letters, they said, blue jeans, the world's single most popular garment, are an indicator of the widespread burden of anthropogenic pollution by adding significantly to the environmental accumulation of microfibers from temperate to Arctic regions.
So this is a good time for me to let everyone know. So I know that I'm usually the one who's saying shower, wash, but actually denim isn't really supposed to be washed very often. Maybe like every few months, maybe just spot wash. I don't know.
But it is true. All of you might think that you're getting all of the lint out of your lint trap in the dryer, and those are all the fibers that are getting out there. But what's happening, and I think we've talked about it on the show before, is that little tiny fibers get into the wastewater from your laundry, and those fibers make their way out into the waterways and are very obviously getting distributed around the globe. So they're finding fibers from all your clothes out there. But hey, because jeans are the most prolific garment in the world, everyone loves jeans.
Fieldo in the chat room. So that's why the ocean's blue? Yes.
Ha 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 ha.
It's true. If you read the ancient Greek texts, the ocean used to be crimson. We completely changed. We went straight past purple into
Yeah. So also,
Thanks, Blue
just
Jeans, for giving us a blue ocean. Okay, if anyone is a child listening to this, that is not how the
yeah,
ocean became
Oh,
blue.
it's not
No,
okay.
no.
it's very incorrect.
All right, Justin, tell me about.
From the publications of the Astronomical Society of Australia, tales of a radio telescope in outback Western Australia, which completed the deepest and broadest search at low frequencies for alien technologies. They scanned over 10 million stars.
Wow.
Dr. Chenua Tremblay and Professor Stephen Tingay used the Murchison wide field array telescope to explore hundreds of times more broadly than any previous search for extraterrestrial life. Tremblay said the telescope was searching for technosignatures, powerful radio emissions at frequencies similar to FM radio frequencies that could indicate the presence of intelligent life in space. The Murchison, I'm going to get this wrong every time. Murchison Wide Field Array is a unique telescope with extraordinary wide field of view that allows us to observe millions of stars simultaneously, she said. That's Dr. Tremblay. We observed the sky around the constellation of Vela for 17 hours, looking more than 100 times broader and deeper than ever before. With this data set, we found no signatures, no sign of intelligent life. Professor Tingay said that even though this was the broadest search yet, he was not shocked by the result. As Douglas Adams noted in Hitchhiker's Guide to the Galaxy, space is big, really big. And even though this was a really big study, the amount of space we looked at was the equivalent of trying to find something in one of Earth's oceans, but only searching in a volume of water equivalent to a large backyard swimming pool. We have to keep looking. So even with that 10 million stars looking for this signature, it was just a very cursory observation. So
There's a lot to look at. Yeah.
There's a lot out there. So the current telescope is a precursor, as all technology is a precursor for an instrument that is coming next, which is the Square Kilometer Array, SKA, an observatory with telescopes that are in Western Australia as well as Southern Africa. Due to increased sensitivity, SKA, low-frequency telescope to be built in Western Australia, will be capable of detecting Earth-like radio signals from relatively nearby planetary systems, said Professor Tingay. With SKA, we'll be able to survey billions of star systems, seeking technosignatures in an astronomical ocean of other worlds. Which I think aren't all other worlds astronomical in nature. But anyway, so the next version of this coming is going to be able to really look at like a lot more.
It'll get us into Carl Sagan, billions and billions of stars.
Yeah, I mean, it should be able to. They're saying the new one's sensitive enough to pick up like a FM signal, like from a planet, like if it was just a regular, like even if it's not a very strong signal from a nearby planet. So it's very, very cool that we're increasing this this ability to look for life. But so far, goose eggs, nothing yet. Nothing. It's not aliens out there yet.
I don't want to find aliens in 2020. We have to wait till we're out of 2020 to find aliens because something bad's going to happen.
Yeah, there is.
That's a great point. Let's not find them this year.
Yeah.
Yeah, like they land and they're like, "Take me to your leader," and you're like, "No, no.
That's okay.
We're gonna go hang out for like a few months, and then or maybe I can give you a ride. We can go to somewhere else and we go, but I'm not taking you there. No way.
Well, other ways that we are planning on looking at the universe is with a really big camera. And there is a camera under development currently that's being built at Stanford's SLAC facility. It used to stand for the Stanford Linear Accelerating Collider. I believe that's what the acronym stood for, but it does no longer because they're not colliding things in there anymore. But they do have a lot of amazing technology, and what they are working on is a camera for the Vera Rubin Observatory that will produce images for the LSST, the large synoptic squared telescope, but it's going to be a really big distributed telescope that they are creating. And the Vera Rubin Observatory is the eye of this observatory with the camera. This camera has the equivalent of almost 216 megapixel smartphone cameras. Like if you took 216 megapixel cameras and you connected them all together and then took a picture with all of them together, that's what this camera is doing. It is 3.2 billion pixels in this image. It's massive, massive image. 3.2 billion pixels, 3,200 megapixels. And the images that are coming out of it so far during their testing are of vegetables. They've taken pictures of Romanesco broccoli.
Wow. So it's broccoli all the way down.
It's broccoli all the way down. Yeah. Yeah. Anyway, they are connecting around 9 of these cameras together to make little segments that then get connected together. And all these things have to be connected together within like a hair's width of accuracy to be able to create the final image for the camera. This camera being tested in Stanford will eventually, now that they've tested it and shown it's perfect, it's wonderful, and they work out all the kinks for the images of broccoli, they will ship it down to Chile, where the eventual observatory is going to be completed, which should give us amazing views of the universe.
And they should be able to spice up that broccoli really nicely in South America.
I think so. Yeah. Nice spices. Mm-hmm. Yeah. Little lime. Yeah. Mm-hmm. Be pretty good.
This is Glenn Brady from the chat room saying, interesting side note, the SKA, that square kilometer telescope, when fully operational, will transfer more data over the interwebs than the entire world today. You thought you had online gaming lag now. There would be a lot of data. Yeah.
It could be worse.
Which is also the problem. Like we can have this technology to grab all this data, to take these high resolution images, but we still are working on the ability to decipher it once it's to even manage that size of data, to break it up, to have it even to have the computing power to analyze it. Let alone the human time and power to do so. We're getting to, it's kind of awesome if you think about it. We're getting to choke points or whatever, where our ability to collect data has surpassed our ability to analyze it.
Yeah.
Which
and
is
sometimes to view it. These images from the Vera Rubin camera, the observatory, they require, if you look at them in full definition, the equivalent of 400 4K ultra high def TV displays displayed in full. That's 3,200 megapixels.
But it's a good problem to have. Bottleneck.
Well, because especially since we're going to be stuck inside for the rest of our lives, some people will be able to analyze some data from their home, which will be great.
Yeah, but that's not true.
It's a great problem to have, because the opposite is we have all this ability to analyze, but we have no data. That would just suck. We've analyzed everything and we're just waiting for another pixel to drop to analyze that. This is the opposite problem. We're going to have stuff that we can take pictures and do investigations and then research it for decades to come to find things. It's sort of like we're creating what we've talked about quite a bit on the show, is that basement in the university or in the museum where all the fossils are stored. And people are, 5, 10, 20, 80 years later, still making discoveries in these basements with this collected data. We're starting to do that with our telescopes now, which is really an amazing, fun time.
So many discoveries to make. This is This Week in Science. We are here. Thank you for listening. If you're interested in one of our shirts, mugs, hats, face masks, head over to twist.org and click on our Zazzle link and browse our store. Lots of items and ways that you can support TWIS. Okay, time for our COVID update. Do-do-do-do-do-do.
Yeah, you got it.
We're still at over 1,000 deaths in the United States per week or per day. There's a lot. It's bad. It's not great. But let's talk about risk. Okay, everybody, let's talk about risk. Today, an interview, some tapes of Trump talking to Bob Woodward, a journalist, have Trump saying, just today and yesterday, some startling facts came out. It's not just older, young people too. Plenty of young people. That's what Trump told Woodward on March 19th. March 19th, at the beginning of this pandemic. And in the meanwhile, over several months, it has been a constant message as to how little young adults have to be concerned about this pandemic and getting this virus. Well, there is there's a research letter that was published this week in the journal JAMA, Internal Medicine, showing that young adults can experience substantial rates of adverse outcomes. Yes, they may not become as sick as individuals, as older individuals as often, but an analysis of 3,222 young adults between 18 to 34 years old hospitalized with COVID-19 indicated that 21% required intensive care, 10% required mechanical ventilation, and 2.7% died. The researchers want people to know that young adults should know that everyone, regardless of age, is at some risk of a severe outcome from COVID. So just to know, take it seriously. You may be safe, but you may also transmit it to somebody else who may not be. Obesity, according to the first meta-analysis of its kind, is a major risk for COVID-19 and for hospitalization and problems with the disease. In the journal Obesity Reviews, an international team of researchers pooled data from scores of peer-reviewed papers capturing 399,000 patients. So this isn't a little study. They're pooling things together. It's observational. This is probably not random controlled trial, but this is nearly 400,000 people they're looking at. They found that people with obesity who contracted SARS-CoV-2 were 113% more likely than people of healthy weight to land in the hospital. 74% more likely to be admitted to an ICU, and 48% more likely to die. That's from Science Magazine's news. So there are things that predispose people to higher risk. And it's not necessarily obesity itself that predisposes people. In the article, they discuss BMI very often. So very high BMIs seem to be the thing that leads to an increased risk of hospitalization, more likely to be admitted to a high ICU and to die. But it is factors that go along with that, like obesity increases inflammation. And inflammation is something that is a huge part of how SARS-CoV-2 affects the body. So if you're already struggling with inflammation, that's going to have a deleterious effect. It also leads to a thickening of the blood and a change in the blood cells themselves, which is a problem very often when people are obese and blood doesn't move as easily, or if there is an issue with circulation. So it's not necessarily obesity itself, because a high BMI on its own does not necessarily indicate obesity, because there are many people who have you are tall, you are big, you are muscular.
BMI is kind of nothing.
It's a vague measure, but they're using it because they don't have a better one at this point. And so that's why I'm trying to make this distinction, that there are other health factors that are involved in it, in changing the way that your metabolism works. So just be careful out there.
Obesity makes you more susceptible to other things too.
Yes.
So it's absolutely. Yeah. And now that we know that it's a blood disease, that makes more sense.
Yep. Yeah. So that's all I wanted to talk about for the COVID corner.
Well, there's more news though. There are more.
There's more.
There's more.
Just
risk
wait.
factors.
There's more.
Yeah, this is a leader of the Ukrainian Orthodox Church in Kyiv, Ukraine, spoke up about COVID crisis back in March, calling it God's punishment for the sins of men and sinfulness of humanity. But he didn't stop there. He clarified. He did clarify. First of all, I mean same-sex marriage. This is the cause of coronavirus.
So please tell me he got COVID. Not to be mean, but...
So, of course.
What is this Schadenfreude?
So, of course, he has contracted COVID-19, and through the very real and effective power of prayer alone has remained in stable condition at a Ukrainian hospital. The COVID illness comes as a great shock to the congregation, who had no idea the patriarch was secretly married to a man.
Right.
In the Sturgis motorcycle rally, in one of the Dakotas, perhaps, they drew a crowd of nearly 500,000 maskless bikers in the height of the pandemic. New paper titled The Contagion Externality of Super Spreading Event, the Sturgis Motorcycle Rally and COVID-19, published by ISA, which is the Institute of Labor Economics. This is a German think tank. They conclude that the rally was indeed a super spreader event that transmitted the virus thousands upon thousands of times. One of the authors of the paper, Joseph J. Sabia, who's also a professor of economics, director of the Center of Health Economics and Policy Studies at San Diego State University, thinks the event could have infected more than 260,000 people all across the country.
Talk about super spreading.
Yeah, he and his co-authors estimate that dealing with the fallout from that rally alone would involve more than $12 billion in healthcare costs, again, spread out across the United States. Governor Kristi Lynn Noem, of whatever Dakota that might have been, was so concerned about the safety ahead of the rally that she told Fox News, "We hope people come. Our economy benefits when people come and visit us." The particular Dakota, I guess it was the South one, now it's been clarified, saw a 35% rise in cases post-rally. Counties and other states from which significant number of residents traveled to Sturgis saw an average of a 10.7% increase.
That's substantial. That's not nothing.
And that lady got COVID, right?
No, not yet. But
I don't want anyone to get COVID, but the people who think it's nothing, it just feels appropriate. That's all I'm saying.
So here's the,
I don't want anyone to get it. Come on.
Here's the thing. 500,000 people. I mean, I get, like, nobody's expecting bikers to be the most responsible citizens amongst us. I think maybe on the road, they follow the road rules very well. They do a great job. But that large of a crowd is coming from across the country and then going back out into the. I don't know that anybody should be surprised by that fallout from the event.
And I do have to add the observation that I've seen people talking about, well, I haven't heard anything about cases rising because of protests, or I haven't heard anything because of compared to
yeah.
Sturgis. And so I think that we
Right.
need to make this point here about indoors versus outdoors and mask wearing.
Right. So actually, this same think tank had looked at exactly what you're talking about previously. They had previously looked at were the mass protest super spreader events, and for exactly what you just stated, the fact that they were outdoors, that there was some social distancing at a lot of them, but that people were all wearing masks. The vast majority of you look at those people. I could just say majority. It doesn't have to be vast. There's no difference between saying the word vast ahead of a majority any more than there is saying vast in front of wasteland. They mean the exact same thing. People were wearing masks. And so those two combination of things, what prevented those from being, there was no evidence of super spreader events from the protests. Because of that, in Sturgis, they were not wearing masks. They were not social distancing. And on top of everything, they were having fun.
No fun having anymore.
That's the thing the pandemic likes least of all is if you are enjoying something, it's wrong. That's how you can tell at this point.
No, it's because they were going indoors to restaurants, bars, cafes in Sturgis and congregating indoors. Yeah. Oh, what a week of news. What a week of news. Did you have one more story?
Yeah, that's right. It's so this is on a serious note. There's another group that was looking at who is doing this. It's a group of researchers from Syracuse University. They were looking at the average daily increase in rural COVID-19 mortality rates. This is people dying, and they found significantly higher rates of death in rural Black and Hispanic communities. So people tend to think of rural America as being just full of toothless white people. People have good dental everywhere now. Thank you. But it's actually 20% of our racial ethnic minorities are in rural areas geographically. And what it was is compared to rural counties with the lowest Black populations, this is the bottom quartile, rural counties in the top quartile of percent Black have a 70% higher daily increase in their COVID-19 mortality rates. And the same thing, applying it to Hispanic populations, ones with the lowest versus ones with the highest. The Hispanic rural areas have a 50% higher average daily increase in their mortality rates. So the research team is not exactly saying what the problem is, but is suggesting that we need to pay a lot more attention to the way healthcare is being distributed in America. And that there seems to be, they're not saying it, but I will, a systemically racist sort of seemingly application to the way that we deal with us and who we see as us. Yeah. From political, governmental spending, where those dollars go, and how the prevention has been or not been taking place.
All right. That does it for our COVID update. This is This Week in Science. Woo-doo-doo-doo-doo. You want to help TWIS grow? Get a friend to subscribe today. That would be fantastic-a-rooney or something. Hey, Blair.
Yes.
Hey, Blair. Hey, Blair. I think it's that time of the show that we love to call Blair's Animal Corner. It's your turn now. Let's hear it from Blair.
She loves our creatures, bright and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
Oh, Justin's not here to say it. Say it, Kiki. I can't start.
What you got, Blair?
There we go. I can't start without it.
The same way.
Where'd he go? Hey, so I have a lovely story about one of our favorites, naked mole rats. So this is a study from the University of Illinois at Chicago, and we've already discovered that naked mole rats can survive acid and live where there's low oxygen content and survive cancer and all this other crazy stuff. But in a new twist, something they're actually really terrible at is hearing.
Oh, really?
They have terrible, terrible, terrible hearing. So they're from East Africa. They're hairless mammals. They're bald. They're wrinkly. They have buck teeth. If you have not seen them, search naked mole rat. You have to search naked mole rat because mole rats are also a thing.
It's a different thing.
They just look like little furry things with giant teeth. But
they don't look furry.
Normal mole
naked
rats
ones. Yes,
are furry. Yeah. So these guys are not furry. They live underground. They live in this crazy society that's kind of like a bee society. They have soldiers, they have workers, they have a queen. They use chemical signals to know who's coming where, when, and what their social rank is. And the higher ranked individual climbs over the lower ranked individual in the tubes or in the underground. I can talk about it forever. But this study is about their communication because most of the communication they do is vocal. So as I mentioned, they use chemical cues too, but they do a lot of squeaking at each other to decide where to dig, how to defend the colony, how to convey the location of food, how to take care of babies. They're constantly chirping and squeaking. But if they have terrible hearing, what the heck is going on here? So this new research found that they had hearing that was so terrible, and then they used technology like used for human hearing. So they performed an auditory brainstem response test. They had electrodes on the scalp that pick up signals that indicate sound being processed by the brain. And the signals were so weak. They have poor hearing. They have hearing so bad that in humans, hearing on that level would be suggested to wear hearing aids. So, yeah.
So they need hearing aids. Okay.
Right. So they're animals that communicate via auditory response, and they have terrible hearing. So why is this happening? So then they looked at the genes. They found 6 mutations in genes associated specifically with hearing loss in humans. So they have 6 independent mutations showing, like, pushing them towards bad hearing. Mm-hmm. So it looks like they're actually being selected for, and they're adaptive, they're beneficial in some way to have this hearing loss. Additionally, they found
what else do the genes do? That's what I want to know. Okay,
yeah, that's a great question. I do not know. But they also found out that the naked mole rats do not have cochlear amplification, which is for humans that don't have the little hair cells in the inner ear or cochlear amplification abilities. People now can get implants to help them do that and hear, and they pretty much can't hear without it. So cochlear amplification is pretty essential in human hearing. So they have these inner ear kind of hair cells, and they send signals to the brain. And without these cells, hearing's really hard. And this is why, like, hearing loss in humans is kind of, it degrades over time. It's progressive. I've been to a lot of rock concerts. My hearing's harder and harder is because loud sounds actually kill hair cells. So they can't regenerate. But these guys, they don't even have them at all. So what the heck is happening here? They actually found that the decibel level of these chirps and squeaks that these mole rats are using to communicate with each other are so loud underground that if they had functional cochlear amplification, they would be lethal to the hair cells. They'd lose all their hearing anyway.
That makes sense. So this is the first thing I was thinking is like, if they all have squeaky voices, maybe they're just trying to tune each other out.
Yes.
So they don't have amplification because they really don't need it.
But okay, but here's the question then.
They're always at a rock concert.
But what came first? Like, did they have to start shouting louder and louder because nobody could hear them? Right. And so they developed a louder squeak over time because nobody could hear them if they just talked in their indoor voice.
So that's possible.
Or the other way.
I feel like that the only way that would make sense is if, as Kiki mentioned, these genes are linked to something else. Because otherwise, why would you get 6 specific mutations to impact hearing before?
Well, so here's what I'm pulling from. I have this cousin who I noticed at a very young age as we were growing up was just so much louder than the rest of us. And I'm loud. I have a loud family. We're all loud. She was a decibel or 10 above everybody else when she talked all the time. It was just her indoor voice was shouting across a very, very long distance. And it turned out she had some hearing issues. So that's kind of what I pulled from it. And I'm thinking like, wow, maybe it was just the fact that their hearing was, through some random mutation that these were like failing genes. And then it starts and then those who hadn't experienced this yet, perhaps, they were just killing it off anyway. So why spend the energy growing these dang hair follicles if everyone else is yelling all the time?
So I think here's what we need to do. Excuse me, University of Illinois. Excuse me. It would be great to do these studies on non-naked mole rats, just normal reg mole rats, mole rats, and then on other rodents, and to see if there's anything indicative in the genome that any relative would have any similar mutations or anything leading towards that, but also to check the decibel level of the squeaks of those other animals. Because, for example, normal mole rats, they go to the surface sometimes. These guys don't really. So they go to the surface a fair amount more. And so they might use, I don't know a lot about mole rats, but they might use alarm calls. So they might be capable of really loud noise.
Or they might also be, since they're going to the surface, listening for predator calls.
So there might be a clue in the chat room. I don't know if it's true, but Timid Tenor says, I remember hearing something about moles using squeaks to freak out worms. Worms scoot to the surface trying to run away from the loud squeaks. I don't know if this is naked mole rats, but this is that would be
not just moles.
Yeah,
but that would be kind of funny that like you used it to get your prey and then it affected your own ability to hear. That would be kind of that would be a catalyst. You need to do this to eat. Would be an interesting evolutionary catalyst for the loss of the hearing then.
And I think that's why doing a comprehensive look at decibel levels across rodents and then additionally looking at these genes and seeing if there's any artifacts, anything similar in any other genomes, that would kind of tell us which came first. But it seems like, I would guess, these guys moved underground. The sound is super insulated now. Like they're pretty much 100% of the time underground. Super insulated. That makes it super loud.
But why wouldn't they just start whispering?
I don't know.
Use your indoor voice.
That's
Use your
a good
underground
question.
voice.
Why are they?
But maybe they
whispering?
How far do they travel from each other? How far do those calls have to go? I mean, there are all sorts of things that need to be asked here. I do believe.
Yeah. So anyway, point being, based on all of this research, they actually think that mole rats might be a good animal model to investigate hearing loss in humans. Because some of the genes that are similar, they're the same genes related to hearing loss in humans, 1, and 2, that this loss of the inner ear hairs is a problem that humans have. So you could potentially look into treatment through the naked mole rats.
A quick poll popped up in the chat room. Caroline Benoit is asking, group poll, if you had to choose between your hearing and vision, which would you choose?
That is tough.
That's a tough one.
So, because I love this show so much, I'd probably say hearing, because then I could listen to the show. But then it would be really hard to prepare the show. So then I don't know.
Just have to get a really good speech to text software.
I have that, but I still have to highlight the text, which is still the problem.
Right, but if you were hearing impaired, you'd have tools.
You know what? I think I would probably yes. It's that's a really tough one. I think I might have to let ouch. I don't know.
Yeah, Goldizator, that is very rude. Does Blair's vision count as having vision? Listen, I might be nearsighted. I might have a terrible astigmatism. I might also be colorblind. But come on now. I can see some things when I wear my contacts.
You can see some of them. Anyway. All right. What's your next story, Blair?
Just a really revolutionary study telling us that, this is from University of Queensland, telling us that if we have protected areas, it helps save endangered species.
What? Interesting.
Mind blown. No, there's a reason this is actually an important study. So we know that we have set aside protected areas in the wild to help animals not have as much pressure from human development and hunting and all these other things, and that they're essential to conservation. But despite this intuitively making sense and the success to this point with those areas, their popularity as a go-to conservation tool comes and goes. And right now it actually looks like it's starting to wane. So there's this increasing debate around global protected areas and whether it actually helps with threatened species. And I think actually we did a study, I think not too long ago, who knows, maybe it was 3 years ago, but we did a study on the show where they looked at protected areas. And as would make sense, if you have a protected area that is small, that is not connected to other protected areas, it doesn't do diddly. Because you can't tell a rhino, just stay over here in this half acre, just stay over, don't go over there. So that is part of the problem. And so I think there's some debate over how best to do this. But we also know that Blair likes to orate about how you can't save species, you can only save habitats. And those habitats save all the species that depend on it. You can't say, I want to save rhinos and just save the rhinos. Really, the only way to save the rhinos is to save the space, their habitat that they depend on. And the positive there is all the other animals that use that space also get a place to be saved. So saving habitats is key to species conservation. But they have to be well funded, they have to be protected, and they have to be well placed. And they have to be big. So these are all like very big caveats, right? But this study looked at the efficacy of these areas and found that they are, in fact, very effective overall. So 80% of mammal species that were monitored in protected areas in this study have at least doubled their coverage in those protected areas over the last 50 years. So their time spent in those areas has doubled. So that means it's helping. They are choosing to spend time in those protected areas. And 10% of those species that they analyzed lived predominantly on that land. That was their main space. So yes, they're very smart. They figured out where it's easier to live. And in these cases, it's easier to live in those protected areas. They looked at 237 threatened terrestrial mammal species from the 1970s today. They measured changes in species ranges and overlaid them with protected area work. And specifically, they did, as I mentioned, they looked at greater one-horned rhinos, Rhinoceros unicornis. So good.
Is it
now,
really?
Yes, it is. Yeah. Which is now, 80% of its range is now in a protected area. So that's something, it's working is basically the point. So without protected areas, some species like rhinos, like tigers, like mountain gorillas, they wouldn't have stood a chance. And so we gave them a space to hide out, to feel safe, to lower their guard. These are all important things, right? And so if you can recognize that, and instead of saying that these two studies, that the study I mentioned previous, that are on opposite sides of this argument, they're not. they're actually both contributing to information that could help save species. We need better funded protected areas. We need more protected areas. And most importantly, we need corridors between them.
Yeah.
Which is really the thing.
So then they can move around.
Right. If we know that animals are choosing the protected area, that's great news. That means if we put a corridor between two, they're not going to walk higgledy-piggledy all over the place. If they recognize that as a protected space, over time, they will choose that corridor. So that it's
possibly.
good news.
Possibly. The first reaction I have to this is those are the protected areas, and so the animals that are staying in those protected areas might be the less wandery inclined, because the animals that were more likely to wander have long since been killed because they left a protected area. And it made me immediately think, like, in a decade's time, as this pandemic progresses, it could be we're left with a planet full of introverts. And that all the extroverted people who like to go out and socialize and just can't help themselves all get felled by the virus. But it made me sort of think, like, maybe the animals that are just like less explorative in nature are the ones that are still left there.
They'll survive humans.
They'll survive the humans, exactly.
Yeah. So it makes me wonder, though, if the corridor thing if they would really take advantage of this versus
so it does
just
work.
staying where they are still.
This is something that's currently in practice. Yeah. So like in South America, they have these poles with a wire over them that go over freeways so that monkeys and sloths can cross above a freeway and not get hit by a car. In California, we have salamander tunnels that go under Highway 101 that the salamanders use. So animals will avoid the dangerous humans if they can. If they're given the opportunity, most of the time, they want nothing to do with us. So we're binocular vision, bipedal, scary stuff. Stay away.
And we have cars and weapons and things, and
So anyway, this is showing us that we're doing good work here. The focus on habitats and protected areas is working, and this is important science because this is the science side of conservation, right? Making sure it's actually working instead of
I think
putting money into things.
It's one of those things that bears repeating.
Checking that it's really working, because the thing that stands out to me is that so many of these species have lost more than 50% of their range since the 1970s. So this is all they've got left. And so we need protected ranges to work. We need something to work if we want to conserve them.
Yeah. So, make sure that they don't get put a pipeline through it or new development on top or anything else. Keep them is basically what we're saying. Keep them. We need more, actually. So, and that goes to marine protected areas and stuff too. All of it.
More.
Don't go.
This is This Week in Science. If you're just tuning in, we just finished Blair's Animal Corner, and we've got a little bit more science to come. Thank you for listening to This Week in Science. Really appreciate the fact that you're here with us this week. And, every week we bring you this show. We try to bring you good science and good conversation around the science, try and delve into issues with curiosity and spark your own curiosity. And it's with your support that we can really do this. With your support, we're able to keep bringing you TWIS week after week. And if you're interested in helping continue to support TWIS and keep us going, head over to twis.org, click on our Patreon link, and choose your level of support. $10 and up per month, and we'll thank you by name at the end of the show if that's what you'd like. Be a part of sharing science and maybe a more sane perspective with the world. Thank you for your support. We really can't do it without you.
Oh, yeah, and we're back with more of This Week in Science.
I was waiting for you to do it. Is he going to do
it? I didn't do it. A fish out of water is a floppy, gasping mess of a creature.
Unless you're a lungfish.
A mud skipper.
Yeah.
A couple exceptions to this rule. Another being the blind cave fish, Cryptotora thamicola, also known as the cave angelfish, which is of the loach species, which lives in caves. This one, with fast-moving water, can be found climbing cave walls at times, going from one place to the other. In a new study, research analyzed the bone structure of this and nearly 30 other hillstream loach species fish, describing for the first time 3 categories of pelvic shapes. Based on the shape of the bone that connects some loaches' spines to the pelvic fins, the team found that there's 10 other species of the loach that shared the cave angel fish's unusually hefty pelvic girdle. This is quotey voice from biologist Zachary Randall. Fishes don't usually have any connection between their spine and pelvic fin. But before the idea was that the cave angelfish was totally unique. So it developed this girdle of pelvic bone on its own. What's really cool about this paper is it shows with high detail that robust pelvic girdles are more common than we thought in the hillstream loach family, meaning that when this blind cavefish ended up in this cave, it was maybe pulling from a previous adaptation that allows this species to adapt to something like this environment. It says that though more than 100 species of hillstream loach are found throughout Southeast Asia, the cave angelfish is the only one whose walking capabilities have been observed and studied. It has this salamander-like wiggle, powered by enlarged ribs, bolstered with stabilizing muscle attachments on its fins, which were first described in scientific reports just in 2016. Randall says the cave angelfish's walk is a key adaptation for surviving fast-flowing cave streams. It can grip rocky stream beds and move between habitats, even up waterfalls as water levels fluctuate in the dry season. Cave angelfish's increased mobility can help it access well-oxygenated stream regions with few or no occupants. Still, little is known about the species, including what it eats. And this is also something very fascinating. It's a blind fish, so it doesn't have vision to find
how's it climbing?
prey.
Doesn't know where it's going.
Well, it doesn't have vision to find prey. So usually when that happens, those fish are using some sort of chemical signatures in the water to track prey. Because they're living in these fast-moving streams where water's constantly going past,
in the dark,
they don't also have what would normally be considered like this pool where these chemical signatures are allowing them to track down prey. So not only do they not know what this thing eats, they have no idea how it would find anything to eat. It's missing all of those things that they assume have found in the past allow blind fish to find prey. So blind or fish that live in very low light environments even. So having the combination of no sight and fast moving water, this is a very confusing fish. And it can come out of the water. So but yeah, it says they converge. This is lead author, PhD candidate Callie Crawford. These loaches have converged on a structural requirement to support terrestrial walking that's not seen in other fish. The relationship among these fishes suggests that the ability to adapt to fast-flowing rivers may be what has been passed on genetically over time. So, yeah, it's a huge mystery fish.
A big mystery.
It's a very Randall and his team most recently observed the cage angelfish on a 2019 cave excursion in northwest Thailand. They were surprised because it's so rare. They found 6 of them clinging to the bed of a fast-flowing shallow stream among glittering stalagmites in one of the cave's chambers.
Okay, so listen. Amphibians came from a fish ancestor.
Right.
So, in the hundreds of millions of years since that occurred, nobody else tried it? Really? This is what I'm not getting, is like, especially considering amphibians can't go everywhere fish can go necessarily. there are unutilized niches that would require getting out of the water. It's crazy to me because like fish out of water, that's the whole thing. Fish are fish, and amphibians are amphibians, and fish don't go on land, and fish don't walk, and all this kind of stuff. But that's where they came from.
Yeah.
So just know.
I mean, we have like the mud skipper is an insane creature. It has like a water lung. Basically, like it does the opposite of what you do when you go underwater swimming. When you take a big gulp of oxygen and you go under the water, it takes a big gulp of water and then comes out of the water and can sustain itself for a time before having to go back and refresh. So there are strategies that, yes, absolutely mimic different progressions out. I like the fact that they described its waddle, though, like you're saying, like a salamander as it's living underground. It's found a similar motility to something that's been very successful at it.
It's because of their pelvis.
The pelvis connection.
It's the same.
It's either the wiggle in the walk or the giggle in the talk.
Oh my goodness!
You got it. That's a moray, right? Is that no?
No, no, Blair.
Missing link has been found between modern day gibbons and its ancient ancestor. This has been found in India, which is sort of interesting because gibbons in Africa are thought to have originated in Asia, I suppose. Maybe it's the other way around. I guess it would be the other way around. But they found this one in India in like sort of the halfway point. That is Asia, I guess it is, isn't it? Hasn't always been. Anyway, this is a 13 million year old fossil. It's from an entirely new undiscovered ape, which is now the earliest ancestor of the modern day gibbon. The discovery by Christopher C. Gilbert from Hunter College fills, according to this, a major void in the ape fossil record and provides new evidence about when ancestors of gibbons migrated to Asia from Africa. Yes, of course. This fossil isn't much. It's not much. It's just a tooth. That's all they have. It's a it's a lower molar.
Not much to talk about, except there is.
Well, there's a lot to talk about just based on this one tooth. It belongs to a completely unknown genus and species, which they named Capyramnagarensis. This is a first new ape fossil of a new ape discovered in the what is a pretty famous fossil site at Ramnagar in India in nearly 100 years. So they've identified lots of ancient apes, lots of species from here, but this is the first one in 100 years, and it's the first gibbon ancestor. Gilbert and team members were climbing a small hill in an area where a fossil primate jaw had been found the year before. While pausing for a short rest, Gilbert spotted something shiny in a small pile of dirt on the ground. So he dug it out and quickly realized he'd found something special. Quotey voice, we knew immediately it was a primate tooth, but it did not look like the tooth of any of the primates previously found in the area. From the shape and size of the molar, our initial guess was that it might be from a gibbon ancestor. But that seemed too good to be true, given that the fossil record of lesser apes is virtually non-existent. There are other primate species known during that time, and no gibbon fossils have previously been found anywhere in Ramnagar. So we knew we would have to do our homework to figure out exactly what this little fossil was. And so this discovery of this fossil was in 2015. So it's been 5 years of studying and analysis and comparisons that's been conducted for them to finally come to the ultimate conclusion that their first guess was correct. It is a gibbon ancestor.
I mean, I guess it takes 5 years to build an entire animal out of a tooth.
Yeah. Let's see. So, yeah. So, there it is. Ancestor of the gibbon. This is published in the Proceedings of the Royal Society B, if you have interest in pursuing.
I love science science like this where it's a tooth and they can identify because of the shape of the tooth, ridges on the tooth, aspects of the tooth, the morphology of it. Okay, this is a gibbon, but this isn't a modern gibbon. This is an old gibbon. This is a gibbon that we've never seen. I mean, the idea that they can put all of the things together is, I mean, the scientific forensics at play are astounding.
Yeah. So this is also sort of it's also showing a migration of apes into Southeast Asia. That's clarifying. So there are gibbons and orangutans in Southeast Asia, but they found orangutan ancestors, I suppose, in this area as previously. But so now this is sort of at the same time, they're also now finding gibbons, the first example of a gibbon in this area. So they can sort of see that they all were transitioning together, that it wasn't just the orangutans here and then somehow a gibbon ended up there much later. They're showing it's sort of a natural radiation that took place. Yeah.
Radiation is good. I mean, not when you're in outer space, but radiation leads to mutation. Mutation is good. Yeah, mutation is great. Mutation maybe leads to genetic change and the change in your abilities. I've got some stories about abilities. One of them is an ability that I do not have. I don't have the ability to break down lactose. You would think with my incredibly Northern European heritage that would be something I would have inherited. But no, I'm one of the few from the North who did not get the particular genes involved in digesting lactase, which is the sugar in milk. Well, a team from Stony Brook University, they looked at some bones from Bronze Age skeletons from a battle that took place around 1200 BC. Their findings looking at these bones and the genes in the bones that indicate lactase persistence and the spread of the ability to digest lactase are published this week in Current Biology. Nobody, really, humans are kind of special because we can drink milk as adults, or at least there's a subset of our population who can. And that's not always been the case. And at one point in time, we did not have the genes to digest milk, but then farming spread through Northern Europe and people started drinking goat milk, cow milk, all the milks, and not just in childhood, but into adulthood to get nutrition. And so there was a spread of lactase persistence as a result of this spread in farming. But they thought it took place over several thousand years, and they looked at this particular pile of bones. They had soldiers, probably about 4,000 warriors from this battle on the banks of Tolens in Germany. And it's a really big battle that occurred in the Bronze Age. A quarter of those 4,000 warriors probably died. So these bones are 3,000 years old. They got the DNA, and the researchers looked into them and found that most of them did not have genes for lactase persistence. So that means that if you look at the gene bank from 2,000 years later in the medieval period, so basically it went from nobody, 1 in 8 warriors had the genetic variant, to almost everybody, over 60% of adults able to drink milk.
So is this going to be mutation or is this going to be natural selection? I mean, is it going to be that if you couldn't handle the new high energy food source that was showing up in these agricultural villages, are you going to be more sickly, less likely to, who's going to want to marry you if you're flatulent every time you simply have something to drink.
But that's part of it. And so the researchers think that the only way that this would have spread so quickly in a matter of 2,000 years, from almost nobody to almost everybody, that in 2,000 years, that it had to have been positive selection. So a case of the genes for lactase persistence positively affecting survival in some way. They don't know exactly what that means, but it could be that maybe parents who did have the genes were gaining enough nutrition to be able to have stronger offspring who were able to survive better. Maybe there was some nutritional benefit or a benefit that kept their immune systems stronger. Was it something allowed them to survive better? And that's what they think happened. They don't think that there would be any other real explanation other than something tied to a positive selection drive during that time period that would make it happen so quickly.
Yeah, and it's something else to keep in mind whenever we're talking about the ancient past. Like whenever you see these movies, the 300, the Spartans, these warriors, these Vikings, these whatever shows, they show these very big, strong, muscular people going to battle. So to be a superior specimen of physicality in the old world basically just meant that you weren't malnourished. Because most people had horrible diets that couldn't sustain muscle building. Like nobody was lifting weights in the old world. That was ridiculous. So when they talk about these heroes, they weren't giants. They weren't super muscular, strong, fit people, right? These were just people who had drink milk occasionally and were like,
maybe got to eat some meat. Here we go.
Had to eat meat occasionally and weren't like spindly and bent over from malnutrition and broken at age 20. Like that's all it took to be a superior physical specimen warrior in the old days was like having decent meals once in a while. So that just drinking milk could be that big separation between a fit society and one that is easily conquered because all you've had to eat is rice for a century. It's just, yeah.
I'm just thinking about how the barter system and dowries and things like that, based on goats and cows and stuff, would not be very helpful if you could not drink milk.
I give you 30 goats to milk for your dowry. I'm not going to let her anywhere near it because I'd like to be. Yeah.
But it is interesting that it has stuck around and it hasn't, the genes have not kept up. Probably has to do with modern agriculture and the spread of the availability of food, but the genes have not kept up their spread globally. Still definitely a large number of people have lactase persistence, but a very large number of people do not. But
you could expect that number to increase for those who don't have it, because I don't feel like we have an agricultural-based society that is expressly reliant on any given food source at this point.
Right. Exactly. Yeah.
Those people are no longer going to be drummed out of society for not being able to eat ice cream.
No, you can't eat ice cream. Get out of town. You go live in the ocean. All right. My last story for the night has to do with surviving the cold, cold nights. We've talked before on the show about torpor. Blair's talked about torpor. We've talked about it's not hibernation, it's torpor. It's the body's metabolism decreases, the heartbeat is still going. It's not quite the same decrease in temperature that hibernation entails. There are some birds that do it. Hummingbirds are among them. And a new study out of University of New Mexico in Albuquerque, researchers have looked at a number of, I think they have 26 hummingbirds from 6 different species. Yeah, that's what it is. 6 different species of hummingbirds that they collected in the Andes, the researchers wanted to know how different hummingbirds use torpor at different higher elevations. So they're up in the Andes where temperatures get really cold. They collected a bunch of hummingbirds and then they put them in little enclosures near the campsite and put a little temperature collecting wire up their cloacas to see what would happen. Because how else are you going to record temperature?
That's science, man.
It's science. You got to put a wire up a bird's cloaca. Anyhow, this wire tracked the body temperatures overnight, and every species of the 6 that they had went into torpor. One species, they had a number of different sizes. One of them was the giant hummingbird, which is about double the size of the 12-centimeter long metal tail hummingbird. The metal tail hummingbird, its body temperature dipped to 3.3 degrees Celsius, which is about 37 degrees Fahrenheit. Almost to freezing. Yes.
No.
Yes, it is the lowest temperature that has been recorded in a bird, in a non-hibernating animal.
So what's interesting there is that's like a temperature if you were going to take a bacterial sample and you want it not to grow. You're not going to freeze it. You're not going to kill it. You're just going to put it in cold storage so that you put it into a G0 state where zero growth is taking place. You do that under about 40 degrees. You do it at about where this bird has put itself, which is very interesting.
And the birds, they look as though they are dead. They don't move. You can pick them up. They don't do anything. They're just kind of dead. But then in the morning when the sun comes up, they warm back up again and they fly away and they are happy and they think about their alien abduction and how strange that was.
So overnight this change happens? Because
overnight.
I think about the extreme temperature changes in bears, because they're like the obvious example, right? And they're lowering their temperature steadily for a while, and then they stay at that lower temperature for weeks to months, and then they raise it back up. It seems like it wouldn't be beneficial to spend all the energy to push it back up for only one night. That seems crazy.
It does, but it is what they are doing. It is an overnight change. They drop that substantially. Some of the hummingbirds do not drop. I mean, the majority of hummingbirds didn't drop that substantially. The average was reaching body temperatures of 5 to 10 degrees Celsius, which is still not, it's not that low, but it's significantly lower than when they're active. And the interesting thing, though, in terms of the trade-off, in the Peruvian Andes, the birds are going to be roosting up in a tree, up on in a crack on a ledge. They're going to find a place to hide for the night and not move. But there are not a lot of prey species or predatory species that are going to come and take advantage of it. So they may have sufficient food and nutrition to be able to support the metabolic costs. And since they don't have the predatory cost, then it might be an adequate trade-off. I don't know, but it's pretty amazing. This is the most extreme hummingbirds. They're extreme, really dropping their body temperatures. And now, we've talked every time we talk about what a cool animal is doing in extreme situations, how can we now apply this to humans? How can we apply understanding how they drop their temperature and raise it back up again with no physiological negative effects that we can see? How do they do that? And how could we do that for, say, surgeries or for, any kind of healthcare procedures that we might need to use it for? Yep. In the chat room, Goldizator is asking how much energy it would take to maintain temperature all night for something so small. And that's actually a very good question because it could be that is the trade-off because hummingbirds take so much energy to maintain their metabolism.
Right. So if you think
because
about
they're so small,
so
it's
a
easier
bear,
to just lose that heat.
Right. So a bear is really good at conserving heat because they're big. So their surface area to mass is it allows them to conserve energy. But yeah, a teeny tiny little hummingbird, their surface area to mass is very different. So yes, they're going to lose heat a lot faster than a bear would.
Yeah.
But it still is a huge metabolic undertaking to push your internal body temperature back up after going into torpor.
Yeah. It's amazing. Hummingbirds, these little teeny 12 centimeters long. Like that's tiny. The tiny little animal is like, I'm gonna freeze. Okay, I'm fine now. Okay. It's amazing. That does it for me for stories. Does anybody have anything else they want to talk about? Nothing else. All right, everybody. If you have questions for us, you can ask questions. We will take your questions and try to answer them. If you have questions, send them to me on Facebook, facebook.com// This Week in Science, or Kirsten at thisweekinscience.com for email. And we will answer your questions on the show. We'd love to get your questions. I think that does it for us. Does it do it? It does it.
Yes,
It
I'm
does.
hoping for a less pumpkin spice sky tomorrow. Fingers crossed.
Yeah, fingers crossed for everyone on the West Coast who is affected by the fires. Yes, let's have a good night. Let's have the wind die down and the flames drop to little embers and homes be saved and the skies return to blue.
Hey, Mother Nature, if you want to throw us some rain, that would be helpful.
That would be awesome. That would be wonderful. Just no lightning, all right? None of that.
Just some rain.
Yeah. Thank you for listening to the show. I hope you enjoyed it. I really, really do hope you enjoyed it. Shout outs to Fada for your help with our show notes and for social media help. Thank you to Gord for helping to man the chat room, making it go, go, go. And for Identity 4 for recording the show. And I would like to thank the Burroughs Welcome Fund and our Patreon sponsors for their generous support. Thank you to Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Maddie Perrin, Gaurav Sharma, Josiah Zahner, Mike Shoemaker, Sarah Forfar, Donald Mundis, Gerald Slurrells, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bignell, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brian Minish, Brendan Minish, Melizond, Johnny Gridley, Flying Out, Richard Reporter, Christopher Dreyer, Mark Bizarros, Artyom, Greg Briggs, John Atwood, Robert, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavis, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Naemberg, Hosty Ranky, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Gary S., Ed Dyer, Tony Steele, Ulysses Adkids, Brian Condren, Jason Roberts, and David Friedel. Thank you for all of your support on Patreon. And if any of you are interested in supporting us on Patreon, you can find information at TWIS.org. Click that Patreon link.
On next week's show, we will be back Wednesday at 8 p.m. Pacific Time, broadcasting live from our YouTube and Facebook channels and from TWIS.org live.
Hey, want to use your ear hairs to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get some friends to listen with their ear hairs too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twist.org. And you can also sign up for our newsletter.
That's right. You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, into the subject line, or your email will go into torpor, fall asleep, and we'll never read it.
Nope. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jackson and Fly, and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This